Hello everyone. So this is a question from NEET 2026. The correct statement with regard to the secondary structure of DNA oblique RNA is okay. Important. What is this secondary structure and DNA and RNA? Okay. So A is DNA possesses a single strand helix and contains uracil as one of the four bases. Okay. So single strand helix and uracil. The second option says DNA possesses a double strand helix and contains thymine as one of the four bases. Okay, so double strand helix and thymine. And C is RNA possesses a double strand helix and contains uracil as one of the four bases. Okay, double strand helix and uracil. And RNA possesses a single strand helix and contains thymine as one of the four bases. Okay. So single strand, helix, and thymine as one of the four bases. The chapters involved is biomolecules. Nucleic acids are a type of biomolecules and they are mainly of two types, deoxyribonucleic acid and ribonucleic acid. Okay. So nucleic acids are of two types, DNA and RNA. Okay. Nucleic acids are also called polynucleotides, very important. So nucleic acids also known as polynucleotides. Okay, so they are known as polynucleotides because the repeating unit in them is a nucleotide. See, nucleic acids are biopolymers, so obviously there will be a repeating unit, right? So the repeating unit in nucleic acids is nucleotides. So therefore the nucleic acids are also known as polynucleotides. So there are many nucleotides which are repeating. Okay, so you can write this down that so the repeating unit in nucleic acids is a nucleotide. Okay, and each nucleotide is made up of three parts. Very important. It is made up of three parts. The first part is a sugar molecule. Second part is a heterocyclic nitrogenous base. Okay, so the meaning of heterocyclic is, so heterocyclic compounds are those cyclic compounds that consist of a heteroatom. Very important. So those cyclic compounds that contain heteroatom. Okay, so I hope everyone knows that what is a heteroatom in organic chemistry. Any atom apart from carbon, for example sulfur, nitrogen, phosphorus, all these guys are known as heteroatom. Okay, so sulfur, oxygen, or phosphorus, anything apart from carbon is known as a heteroatom. So those cyclic compounds which contain a heteroatom are known as heterocyclic compounds. For example, pyridine. Pyridine is this. So you can see, yes, it's a cyclic compound, but also it consists of a nitrogen or a heteroatom in the ring. Okay, so therefore the second part of nucleotide is heterocyclic nitrogenous. That means in the heterocyclic compounds we will have nitrogen in the ring. Okay, and the third constituent of nucleotide is phosphoric acid, which is H3PO4. So these are the three things that makes a nucleotide. Okay, so in general we can represent a nucleotide like this. So we will have sugar, which will be connected to a base, and that sugar is also connected to a molecule of phosphoric acid. So in general, this is a nucleotide. Okay, and in nucleic acid, since a nucleic acid is a polynucleotide, so there will be many such repeating nucleotides. They will be repeating. Okay, so therefore we can represent the nucleic acid like this. So we will have many of such nucleotides: sugar connected to base, oxygen, and then we will have again sugar connected to this, connected to base, is connected to another phosphate group, and it goes on. So this is the representation of a nucleic acid. Okay, so it is made up of many such nucleotides. Okay, and the difference between DNA and RNA is, the first difference is in them, the sugar is different. Remember, I sugar is a constituent of the nucleotide, right? So DNA contains, the sugar in DNA is beta D2 deoxyribose, and in RNA the sugar is beta D ribose. So I will draw their structure. In beta D2 deoxyribose, it will look something like this, and both are pentose sugar, by the way. That means they contain 5 carbon atoms. So OH, H, HH, HOH, CH2OH, and H. So this is beta D2 deoxyribose, and beta D ribose is OHH, HOH, HOH, CH2OH, and H. Okay, so you can see here, let's say this is first carbon, this is second, this is third, fourth, and fifth. So I will give the numbers 1, 2, 3, 4, 5. Here also 1, 2, 3, 4, 5. So you can clearly see this is the sugar in DNA, and it is known as deoxy in the second position because there is no O here, compared to the sugar contained in RNA, which is beta D ribose, which contains O. Then let's talk about the bases. Okay, there are two different classes of heterocyclic nitrogenous base which have been found in nucleic acids. Okay, so one such class is known as purines, and the other class is known as pyrimidines. Okay, so purines, in purines, the two most commonly found purines are adenine and guanine. Okay, symbol is A, guanine symbol is G, and these two are present in both DNA as well as RNA. Okay, so they are found in DNA plus RNA. Then coming to pyrimidines, the three most commonly occurring pyrimidines are uracil, symbol is U, thymine, symbol is T, and cytosine, symbol is C. Okay, so uracil is found only in RNA, thymine is found or occurs only in DNA, and cytosine occurs both in RNA as well as DNA. And the structure of nucleic acids is, discussed at two levels. So the first level is primary structure, the second level is secondary structure. Okay, so let's discuss about what's the meaning of a primary structure of a nucleic acid. Okay, so Primary structure is the sequence in which the four nitrogen bases are attached to the sugar phosphate backbone of a nucleotide chain. Very important. Okay. The sequence in which the four nitrogen bases. So in the case of DNA, the four nitrogen base will be adenine, guanine, thymine, and cytosine. In the case of RNA, it is going to be adenine, guanine, uracil, and cytosine, right? So the sequence in which the four nitrogenous bases are attached to the sugar phosphate backbone of a nucleotide chain. So this sequence is known as the primary structure. Okay, to understand this, I will, draw a nucleotide chain in general. So it will look something like this. Remember, we have already drawn it. Do you remember this? Yeah. So this is a nucleic acid or a nucleotide chain. So let's draw this again. So we will have sugar, which will be connected to base, that will be connected to a phosphate group. So I will change the color. Yeah, sugar and is connected to base, which is connected to a phosphate group, and it continues, right? So obviously this base can be, for example, in the case of DNA, it can be anything out of AGTC, right? Adenine, guanine, thymine, cytosine can be anything. Similarly, this base also can be adenine, guanine, thymine, or cytosine. So this base order, for example, A, G, T, A, C, T. So this is, one example of an order. So this base order or sequence is known as the primary structure. And now let's discuss about the secondary structure of DNA. So DNA consists of two polynucleotide strands or chains, and these strands run in opposite direction, giving a double helix structure to DNA. Very important. Okay. So we'll write that down. DNA consists of two polynucleotide chains or strands, and these strands run in opposite direction, giving a double helix structure. Okay. So to draw this, so let us say that this is one of the strand, and let's say this is the other strand. Okay, so it consists of two polynucleotide strands, and they run in opposite direction, giving a double helix structure to the DNA. And the backbone of each strand, that is this is the backbone. When I say backbone, this is the backbone of the other chain. Okay, so the backbone of each strand consists of sugar phosphate units. Okay, very important. So here we have, sugar molecules which will have base connected to it, sugar base connected to it, sugar base connected to it, sugar and base connected to it, base connected to it, and it continues throughout this strand. So we'll have sugar and base connected to it, and base units of each strand are pointed into the interior of the helix. So for example, if sugar is here, so base is towards the interior of the helix. It is, not like this. It will be exterior to the helix. This is incorrect. The base are always towards the interior of the helix in both these strands. Okay, here also sugar, so base is interior to the helix. Okay, good. Base pairs of two strands are linked together through hydrogen bonds. Very important. Let's say that we have a base here, and let's say that we have another base here. So these two bases are linked to each other by hydrogen bonds. Okay, and purine base of one strand is always paired with a pyrimidine base of other strand. Let's say that this base is purine, so it will be connected to the base of the other strand, but that base has to be a pyrimidines. Very important. Okay, so purine base of one strand is always paired with a pyrimidine base of the other strand and vice versa. Okay, so I will write these things down. Very important that purine base of one strand is always paired with a pyrimidine base of other strand. Okay, and considering all the geometry and the number of hydrogen bonds that they can form, guanine always it is paired to cytosine and vice versa. Guanine is always paired to cytosine and vice versa, that is cytosine is always paired to guanine, and there will be three hydrogen bonds between them always. Okay, and similarly adenine is always paired to thymine and vice versa, that is thymine is always paired to adenine through two hydrogen bonds. Okay, so therefore to understand this, so let's now be specific about the bases. So let's say that I have guanine as the base here, so therefore this guanine will be connected to a cytosine of this strand, because only guanine and cytosine can form a bond, hydrogen bond. So therefore this is connected to cytosine through a three hydrogen bonds. Similarly, let's say we have a adenine of the blue strand, so it will be connected to the thymine of the red strand by a double bond. Let's say we have thymine of the red strand, so it will be connected to the adenine of the blue strand by a double bond, double hydrogen bond. Okay, similarly let's say we have cytosine in the blue strand, so it will be connected to the guanine of the red strand by three hydrogen bonds. Okay, so therefore the two strands of the double helix are complementary and they are not identical. Okay, they are complementary. The meaning is that base sequence of one strand automatically fixes that of the other. For example, if you know the sequence of this blue strand, you already know the sequence of the red strand, because we know that if guanine is there, it has to be cytosine in the other strand. If adenine is there in this strand, we know that it has to be thymine in the other strand. Okay, but unlike DNA, RNA just has a single strand. Okay, so RNA is something like this. RNA has a single strand. So now, knowing these informations, now we can go and see the options. Um, the first option is DNA possesses a single strand. Incorrect. It has a double strand helix structure, and also it contains uracil. No, it does not contain uracil. Okay. Option B, DNA possesses a double strand helix. Correct, and it contains thymine as one of the four bases. Yes, so this is the correct option. Okay, so let us look at option C. RNA possesses a double strand helix. No, RNA is a single strand, consists of a single strand. Okay, and it contains uracil. Yes, it does contain uracil, but it is a single strand helix structure. And the fourth option, that is RNA possesses a single strand helix, correct, and contains thymine. No, RNA does not contain thymine, so this is incorrect. So even D is incorrect. So therefore the correct option is B. Okay, thank you.